Welcome to this Icer meeting. We will be beginning in less than just to let people have time to log in.
Hello, everyone, and welcome to this meeting. Today, we will be discussing the anti-B cell maturation antigen CAR T cell and antibody drug conjugate therapy for heavily pretreated relapsed and refractory multiple myeloma. And we appreciate you all being here with us today. At the beginning of every Midwest CEPAC meeting, we want to ensure that all speakers disclose any financial relationships with industry. Every member of our Midwest CEPAC panel who is present today has met the Icer conflict of interest policy. Would each Midwest CEPAC member introduce themselves, their work, and note whether they have any updates to their declaration. I will start, and then I will call on different panel members. My name is Reem Mustafa. I am an associate professor of medicine, Division of Nephrology at the University of Kansas Medical Center, and I direct the outcomes and implementation research, and I will be chairing this meeting today. I have no conflict of interest to declare. Dr. Alan Balk.
Hi, I'm Alan Balch. I'm the CEO of the Patient Advocate Foundation, and I have no updates to my conflict of interest statement.
Thank you. Mr. Nick Bagley.
Hi, I'm Nick Bagley, professor of law at the University of Michigan with a focus on health law. I have nothing to declare.
Thank you. Dr. Bijan Bora. Okay. Let's go to Ms. Angela Brown.
Good morning, Angela Brown, CEO for the St. Louis Regional Health Commission, and I have no conflicts of interest to declare.
Thank you. Dr. Kurt Busch.
Hi, Kurt Vandenbosch. I'm a pharmacist with St. Luke's Health System in Idaho, and no changes to conflicts of interest.
Thank you. Dr. Greg Kerfman.
My name is Greg Kerfman, a deputy editor at JAMA, the Journal of the American Medical Association. I have no conflicts to disclose.
Thank you. Dr. Stacy Dusitsina.
Hi, everybody. Stacy Dusetsina. I'm an associate professor of health policy and Ingram associate professor of cancer research at Vanderbilt University School of Medicine, and I have no financial conflicts of interest industry to disclose.
Thank you. Dr. Ingvar Falkjader.
Hi, this is Ingvar Falkjader. I'm a professor at Case Western Reserve University, and I have no conflicts of interest to disclose.
Thank you. Dr. Albert Huang.
Hello, Albert Huang, general internist, professor of medicine and public health sciences at the University of Chicago. I have no conflicts of interest.
Thank you. Dr. Jill Johnson.
Good morning, I'm Jill Johnson. I'm a clinical pharmacist and professor of pharmacy practice at the University of Arkansas for Medical Sciences College of Pharmacy, and I have no conflicts.
Thank you. Dr. Bradley Martin.
Good morning, everyone. I'm Bradley Martin from the Division of Pharmaceutical Evaluation and Policy here at the University of Arkansas for Medical Sciences. and I have no conflicts to report.
Thank you. Dr. Tim McBride.
Good morning. I'm Timothy McBride. I'm a professor in the Brown School at Washington University. I'm also co-director of the Center for Health Economics and Policy, and I have no updates to my conflicts of interest statement.
Thank you. Professor Rachel Sachs.
I'm Rachel Sachs, an associate professor of law at Washington University in St. Louis, and I have no conflicts to disclose.
Thank you. And Dr. Stuart Winston.
Hello, everybody. I'm a clinically retired cardiac electrophysiologist working currently in the patient experience domain in a large multi-specialty practice in Ann Arbor, Michigan. I have no conflicts of interest.
Thank you. I'm going to try one more time. Dr. Bijan Bora.
Hi, sorry, I was muted first time. I'm Bijan Bora. I am a professor of health services research at the Mayo Clinic College of Medicine. I have nothing to disclose. Thank you.
Excellent. Thank you all for introducing yourself. We're also very fortunate to have our patients and clinical experts. I would like to ask you to introduce yourself and declare any financial relationships with industry or other potential influences on judgment when you speak. We will also present this information on a slide, I believe, and we will start with Tom Pilford.
Sorry, what is the question?
We would like you to introduce yourself and declare if you have any conflict.
Well, I have no conflicts whatsoever. I'm 76 years old, happily married for 40 years, happily retired. I have had 5 different protocols dealing with my multiple myeloma. I started out, my first induction was Cyborg D. Then I went on to Revlimid. No, I went on to Velcade, Cytoxin, and Dex. Then after that, I went on to Revlimid plus Dex. And after that, I joined my first clinical trial, which was a combination of a drug called Icer with carfilzomib. And when that fails, here I am, a CAR T patient who has survived so far 14 months.
Great.
And I've just had my 2 shots, and then I had a COVID-19 antibody test, and I've proven to be positive. So life is pretty good.
That's great. Thanks for being here.
Sure.
Dr. Anita D'Souza.
Hi, good morning, everyone. My name is Anita D'Souza. I'm an associate professor of hematology oncology at the Medical College of Wisconsin in Milwaukee. My conflicts of interest are shown on the slide. I don't have any more updates to that. Thank you.
Thank you. David Mitchell.
Good morning. I'm David Mitchell. I am a multiple myeloma patient. I was diagnosed a little over 10 years ago. I am currently on a 4-drug regime, a 4-drug combination. And that 4-drug combination includes the 3 primary classes of myeloma drugs. So when I fail on this combination, I'll be triple refractory and a candidate for the drugs that we're going to talk about today. So this discussion is very important to me, and I have no other conflicts to disclose beyond those listed here. Thank you.
Thank you. Dr. Vincent Rajkumar.
Hi, everyone. I'm Vincent Rajkumar. I'm a professor of medicine at the Mayo Clinic. I've devoted about 20 to 25 years of my career to the treatment of myeloma and running clinical trials for this disease. I have no financial conflicts with companies. I'm currently on the board of directors for the International Myeloma Foundation, as well as vice chair for the International Myeloma Working Group.
Thank you very much for all of you to be with us today. I'm going to ask that anyone else who speak, including policy roundtable participants and individuals making public comment, that they should announce any financial relationships with industry or other potential influences or judgment when they speak. And we will always try to present this information publicly on slides when available. With that, I would like to introduce Dr. Steve Pearson, President of Icer, who will take us through an introduction of the day. Thank you.
Thank you, Raymond. Thank you very much to our patient experts and clinical experts who are joining us for the whole day. Let me just give a brief introduction before we really get into the meat of the meeting. I want to make sure that people understand and appreciate the number of people who are on the webinar watching why we really are here today. And I'd like to start with a quote from a patient. There is not a day that goes by that I'm not aware that I have multiple myeloma. I know that while the current combo is working, it won't work forever. I'll then be refractory to all three major lines of treatment. And that means I will have to look at autologous stem cell transplantation and or one of these new classes of drugs which come out with greater toxicities. I know that without new drugs, I will run out of options. So one of the main reasons we are here today is because there is a significant, still unmet need, decades of work on multiple myeloma treatments, a lot of progress, but still a lot of unmet need for many, many patients who will reach a point at which all of the available treatments are not working well, or whether there might be a new treatment that might offer, again, better options with less toxicity. So the patient need is really central to why we're here today. And that also means that the day that these treatments have been or will be approved by the FDA, what happens is a sense of celebration. It's a sense that patients who've been waiting, patients and families who have participated in clinical trials, investing their time and effort and risk. It also means we celebrate the risk that companies take, the investments, the time that they put into these treatments, often over many, many years. And so when that comes to fruition with a new drug available through the FDA, it's a serious time of true celebration. What also happens that first day is that things start happening in our health system. The price gets set, coverage gets set, and patients start to have access difficulties often. And that can come either through aspects of coverage, eligibility that may be uncertain or difficult for them to meet. There may be costs or will be costs, either out of pocket and or insurance premiums that start to become part of the landscape of how we need to think about these new treatments. And so we're here because in some sense, not only are we talking about the patients and the celebration around these treatments, and we need to keep our eye on others in the healthcare system. And just to kind of put faces to this concept, this is a slide from publicly available material of folks who have wrestled with either themselves or people that they care about when they realize that they don't have enough money to pay for health insurance. So Mr. Edlow is a pharmacist himself and a pastor in Virginia, and he has worked over many, many years with members of his community that he sees wrestle every single day with whether they can afford the treatments that they're prescribed, not just the out-of-pocket for the treatment itself, but the rising health insurance premiums that sometimes block their ability to get good care. The Whitmans are a family in Alaska, and this is now from several years ago, but they felt that they just could no longer afford the insurance premiums being self-employed, and they had to give it up. And that they felt put their family at risk, and they knew that. And then both of their kids had health issues. Their son in particular was hurt, and the story was told that they felt he should have gone to the emergency room, but they didn't take him. And so as parents and as a family, they worry about the health effects of the fact that they don't feel like they can afford health insurance. And Mr. Breen on the far right is a small employer in Minnesota who told the story of his needing to give up providing health insurance to his employees in a small business. And he said he watched how they really suffered, not just himself with that risk, but the sense that some had to leave their job that they really liked because they just had to have health insurance, and others, again, feeling like they're under constant pressure and risk not having adequate coverage. So in a sense, we're here today to have a really sharp focus on the evidence and the broader perspectives on value for patients and their families. But we have to remember that around them, around all of us, are other families. And forgive the stereotypical iconographic picture here of families. They come in all different colors, shapes, sizes, compositions. But there are people around us who aren't in this virtual room today. And we just have to think that ultimately we want a health system that can build towards an ability to care immediately and with everything we've got and in the innovation pipeline rich with new things to think about for the patients in front of us. But we have to have a balance. We have to have a way to structure it so that we have the ability to keep insurance affordable for those around us. So that's why we're here today. And you've met the members of the Midwest CEPAC. I need to tell you a bit about Icer, which is the convening organization and the academic secretariat for the report that undergirds the discussion today. First, our disclosure of our sources of funding is updated on our website and easily available, showing not just the percents, but the individual organizations. As you can see, overall, the majority of our funding comes from nonprofit foundations. The chief among them are the Laura and John Arnold Foundation, the California Healthcare Foundation, and recently we've had funding from the Commonwealth and the National Institute for Healthcare Management Foundations. We do take approximately 20% of our funding for a separate Icer Policy Summit program and other activities that are not related to the research and the meetings that we hold. And those contributions come directly, as you can see, from health plans and provider groups and from manufacturers to help support that activity. We have a small slice of government contracts and other funding. And again, this is kept updated on our website at the link shown above. The report today started almost 8 months ago. And it starts with a deep dive that we call scoping, that starts with conversations with patient groups, with clinical experts, manufacturers, and other stakeholders. But the key there really is the patient groups, because we have to go into these knowing that there are elements of the experience, the diversity of the experience, kind of the broader thinking around what value is and what even what clinical outcomes matter to patients that we will miss if we just go out and look at the peer-reviewed published evidence. So we work with all of these folks to try to make sure we get oriented and ask the right questions and include or even generate evidence where it's possible to make a richer, more comprehensive view of value. We take an internal Icer staff team that looks at the evidence and works with these other stakeholders around the possibilities of looking at evidence that may not be in the public domain. And we often work with outside academic partners in the cost effectiveness modeling. In this case, it was with faculty at the University of Colorado, and you will meet them later. The report itself goes through multiple cycles of public comment and revision, both individually with the stakeholders and then through broader public comment. And we were fortunate and want to thank our expert reviewers in addition, who took a very deep dive into the report. And these are Elizabeth Franklin, who's president of the Cancer Support Community, Dr. Raj Kumar, who is here with us today, and Dr. Vij at the Washington University School of Medicine. The evidence in the report itself really does try to, again, match the breadth of these discussions and the views on value, because value itself is comprised of different components, if you will. And the report breaks it down in some sense into these different factors, but ultimately we need to kind of bring them all together. So we look at how treatments and other interventions might help us by providing a longer life. They might give us health benefits by returning us to better function, or by having fewer side effects and other treatments available. When we look at costs as a part of value, we are looking at the broad picture, all the costs in the healthcare system, not just drug costs when we're talking about the health sector perspective. And we also have information around trying to estimate the effects outside the healthcare system as well. The buckets in blue there are in a sense, are potential benefits or disadvantages that extend beyond the traditional definition of health. There will be specific votes taken on the impact of treatments, broader ability to help patients with their life their education, their work, and those kinds of things. And then in some sense, surrounding these discussions, there are often social or ethical priorities that we want to be conscious of and bring to the table around whether there's special priorities due either to the history of this condition, its special burdens on patients and families that we want to make part of the broader view of value. So these are in our report, and they will be prominent features of the discussion today. And so welcome again to this meeting. And at this point, the agenda is fairly straightforward. It will start off with the clinical evidence and economic evidence presentations. We have our patient experts and clinical experts at the table to help with that. There will be a period of public comment and discussion, fairly brief lunch break, and then we'll come back for further deliberation on the evidence and votes. And then we'll end the day with a policy roundtable at which we'll kind of move into a kind of narrow focus on listening and voting to a kind of forward-looking conversation on how does this project into the future? How should we think about clinical practice evolving with these new treatment options? How do we need to think about future research to fill in the gaps? And importantly, what can we learn about best approaches to thinking of coverage and pricing and payment options to make sure we meet that target captured in that picture earlier of being able to do the best for the patients today and tomorrow, while also keeping a broader view of the health system and the communities around us. Let me hand it over then to Dr. Sei Li, who will introduce himself and take it from here.
All right, can everybody hear me? Well, thank you, Steve. So my name is Sei Li. I'm a professor of medicine in the Division of Geriatrics at the University of California, San Francisco. And I was privileged to lead the review of the clinical evidence. Next slide. So first and foremost, I want to thank the Icer research team. They've really been instrumental in kind of gathering a huge amount of data and evidence together and making my job a lot easier. And we have no conflicts of interest to disclose for this report. Next. So as a quick way of background, multiple myeloma is a hematologic cancer of plasma cells affecting approximately 150,000 Americans. The median age at diagnosis is 69, and we will come back to that when we look at the studies as well as the overall impact, the lifetime impact. And importantly, Black Americans are 2 times as likely to be affected than white Americans. Focusing in a little more on the disease, the focus of this review, we're going to talk about relapsed and refractory multiple myeloma. The currently approved therapies are not curative, and so patients usually cycle through multiple combinations of treatment, resulting in significant side effects as well as high cost. And because these therapies are not curative, there are many patients who kind of get to the point where none of the many medications that are available, that are approved, are working anymore. And so there's a high unmet need. Next slide. So I want to start and end really with kind of what I think should be the focus throughout our deliberations, which is the patients. So I want to talk about what the patients and advocates told us about living with this disease. And really the 3 themes that came out was, number 1, kind of how fatigue and weakness really affects every part of their lives. A second theme that came out was how much of a financial strain that this disease sometimes imposes on patients. And the third theme is kind of the impact this disease has on not only the patient, but the caregiver and the family unit. So we'll come back to these later on in the presentation as well. Next slide. So again, zeroing and focusing in on the areas and the medications that we'll be looking at, there are three broad classes of commonly used treatments. First are monoclonal antibodies, specifically daratumumab or isatuximab. Second are immunomodulators, most commonly lenalidomide or pomalidomide. And then the third class are proteasome inhibitors, bortezomib, carfilzomib, or ixazomib. The other terms that I think we all need to be familiar with is triple class refractory. So these are patients whose disease is no longer responsive or unresponsive to each of these three classes of treatments. Then the second phrase that we'll talk about are quad and penta refractory. And as you can imagine, these are patients whose disease is unresponsive to 4 or 5 commonly used myeloma medications, including at least one medication from each of the classes noted above. Next slide. So I want to talk, before we dive into the specific agents as the focus of our review, I want to talk about Bcma, which stands for B cell maturation antigen, and why it is an attractive target for the treatment of myeloma. So Bcma is overexpressed on plasma cells, but appears to be minimally expressed on other cells. And it appears to be essential for the survival of long-lived plasma cells in myeloma. And so this appears to be something, if we could magically target this antigen, then the hope is that we can target the cancerous myeloma cells with little effects on other types of cells. Next slide. So we get to the medications, the new interventions that we'll be focusing on this review. So the interventions fall into 2 classes. One are the CAR T cell therapies, and the 2 specific ones are the idacaptogene viclacel, or commonly called Ida-cel, and now as of the FDA approval, the brand name is Bcma by BMS and Bluebird Bio. The second CAR T cell therapy is ciltacabtagene autoleucel, or cilta-cel, and that's from Janssen and Legend Biotech. The third medication, which also targets the Bcma antigen, is a type of drug called an antibody drug conjugate, and that is the belantamab mafodotin, and the trade name is Blenrep from GSK. And the comparators we'll be using are the combination of usual care regimens. Next slide. So I want to take a step back, because these are not kind of simple pills that we ask a patient to take once a day, and these are fairly complex treatments. What exactly CAR T process looks like, as well as what the antibody drug conjugate, what that is. So here's a very brief overview of the CAR T process from the graphic made by Dana-Farber. So the first part of the CAR T process is leukapheresis, where a patient's white cells are collected. And then there's this manufacturing process where the T cells from that patient are isolated and then activated. These T cells are then transfected with a specific gene that can identify the Bcma antigen. And then once these transformed T cells are again isolated and then expanded so that we have enough of them. And so this is the patient's white cells that have been modified to specifically identify and attack the Bcma antigen. And these white cells are infused back into the same patient. Next slide. So the second type of anti-BCMA intervention was this antibody drug conjugate. And the example that we'll be examining today is the belantamab mafodotin. So the belantamab is made of 2 parts that are linked together. One is the antibody, the belantamab that binds to the BCMA on the surface of the plasma cell. And then the drug itself is the mafodotin that accumulates and kills the plasma cells. So the way that people have talked about this is that this is supposed to be some sort of an analogy as a guided missile. The guidance is the antibody that should just focus the on the BCMA, and then the drug is the payload or the thing that will actually, hopefully, preferentially attack the myeloma cells only. Next slide. So again, focusing down on the scope of our review. So the first part of it will be the CAR T therapies, or ide-cel and cilta-cel versus usual care. And the population there are adults with triple or quad refractory myeloma who have received at least 3 prior lines of therapy. And then the second review will be the belantamab versus usual care. And those are will be focused on adults with triple, quad, or pentarefractory myeloma who have received at least 4 prior lines of therapy. Next slide. The outcomes that we'll be assessing will be overall response, progression-free survival, overall survival, health-related quality of life, as well as the treatment-related harms. Next slide. So I want to, again, as I mentioned, I wanted to start and kind of end with talking about these, what we found from talking with patients and caregivers. And regarding kind of the fatigue and weakness, a representative quote talked about how the weakness is the worst thing. It interferes with your ability to exercise and take care of yourself. Regarding the financial strain, one of the patients that we spoke to talked about that first year I went into debt and I had to refinance my home. I was 3 years from paying off my house and I had to start all over again. Third, regarding the impact on caregivers, one patient talked about, the irritability caused by the dexamethasone, one of the myeloma treatments, led to a temporary estrangement with my spouse because of my short temper. So just to kind of re-emphasize how much of an impact this disease can have on patients and caregivers. Next slide. So now we get into the clinical evidence and some of the numbers that looking at the specific outcomes. So this is for triple or quad refractory myeloma with at least 3 prior lines of treatment. This is an overview of the key phase 2 open label single arm trials. The primary trial for ide-cel is the KarMMa study. The primary trial for cilta-cel is the CARTITUDE-1 study. And the usual care we'll be looking at is the Mammoth Study, which is a retrospective multi-center chart review study focused at, I believe, 11 academic medical centers around the country looking at this population. You can see the relative sizes of each of the studies, as well as the ages and follow-up. The two things, the KarMMa and CARTITUDE studies reported an as-treated response rate. And you see the KarMMa study had a 73% overall response rate, and then the CARTITUDE study had a 97% as-treated response rate. However, we felt like it was probably fairer to look at the intention to treat overall response rate, since that's what was reported in Mammoth. And if we look at the overall response rate for KarMMa, it's 63%, CARTITUDE it's 75%, and Mammoth it's 31%. Again, if you look at the median progression-free survival in the across the treatments, for KarMMa and ide-cel, it was 8.6 months. For CARTITUDE, it was greater than 12.4 months, and that means that they hadn't reached at 12.4 months, the half of the people had not progressed. And then for Mammoth, it was 3.4 months for usual care. And then overall survival for KarMMa and ide-cel, it was 19.4 months. For cilta-cel was again greater than 12.4 months, and Mammoth was 9.3 months. The one thing that I wanted to kind of highlight was the fact that the ide-cel in the KarMMa study did mention that 28 patients were retreated after disease progression, and that's a point that we'll come back to in the modeling study, in the modeling component later on as well. Next slide. So this is the slide looking at the clinical evidence for belantamab versus usual care. And so again, this is the triple, quad, or pentarefractory myeloma patients who have been exposed to at least 4 prior lines of treatment. And the DREAM2 study for belantamab is the key phase 2 open label single arm trial. And the comparison that we have is also from Mammoth, but we specifically chose the parts of Mammoth that were closest to the population in DREAM2. So specifically focusing on people who were at least triple refractory as well as pentarefractory. And so here you see the age and the follow-up duration. The overall response rate, the DREAM2 study did focus on intention to treat and published the intention to treat numbers, so we can provide those. 32% overall response rate in DREAM2, and in the Mammoth sub-cohort was 28% with usual care. The DREAM2 had a median progression-free survival of 2.8 months and median overall survival of 13.8 months. And for the Mammoth subcohort, you see that in the triple quad refractory group, the overall survival was 9.2 months, and in the penta refractory subgroup was 5.6 months. Next slide. So I also want to talk about the patient-reported outcomes, and what this shows is the mean change from baseline in health-related quality of life across each of the 3 trials that we talked about for ide-cel, cilta-cel, and belantamab. So you can see that the ide-cel study, the time from baseline was 9 months after the initiation of treatment. Cilta-cel was 6 months, and belantamab was also 6 months. Looking across the ide-cel results, you see improvements in physical functioning and decreased fatigue, decreased pain, and improvements in global health. And the dagger behind each of those numbers shows that those results are statistically significant. And if you look at one of the footnotes, you'll see that the usual benchmark is that changes of greater than 10 points is considered to be clinically significant. So for ide-cel, we see that treatment has generally led to clinically and statistically significant improvements in physical functioning and global health, as well as clinically and statistically significant decreases in fatigue and pain. For cilta-cel, of note, I think the time from baseline is important, that this was only at 6 months versus 9 months for ide-cel. You see that we don't have results for physical functioning or global health, but for fatigue and pain, the results are statistically significant. Decreases in fatigue and decreases in pain. It doesn't quite reach the commonly common threshold of 10 points, but they're close, and it does look like there may be improvements or decreases in fatigue and pain with cilta-cel. Looking at the quality of life outcomes from belantamab, you see that none of the changes in baseline reach statistical significance, and none of them get close to the widely accepted threshold of 10 points for clinical significance either. So belantamab does not appear to improve or worsen the health-related quality of life. Next slide. So I want to talk also about the treatment-related harms associated with each of these medications. So for the CAR T cell therapy, ide-cel and cilta-cel, the most important toxicity that we should talk about is the cytokine release syndrome. And the rates of it occurring is really kind of, it occurs in the majority of patients, 84 to 95% overall. we looked at grade 2+ as being significant and something that we should focus on, and it occurs in 31 to 39% of patients. And just to give people who are not oncologists a kind of a background, grade 2 is usually means that a patient has hypoxia or low oxygen or low blood pressure, and in the overwhelming majority of cases requires hospitalization at this point. The CRS lasted a median of 4 to 5 days, and 52 to 69% require tocilizumab, which is an intravenous medication that is designed to try to blunt the immune response that is at the heart of CRS. Neurotoxicity occurred in 18 to 21% of patients receiving CAR T-cell therapy, and 3 to 6% had treatment-related deaths. And specifically, the 3% is from the Icer treatment, and 6% is from cilta-cel treatment. Looking over to the right panel for the harms of belantamab, the primary harm was the ocular toxicity and visual decline. And so specifically, 46% had 2 or more lines worsening on the Snellen chart in the more affected eye. And so the Snellen chart is the visual acuity chart that I think many of us go look at when we go to the optometrist. And so this is one eye that is more affected. 46% dropped 2 lines in their visual acuity. 18% their visual acuity in the better eye, so in the less affected eye, got worse than 20/50, meaning that this is a level that oftentimes requires changes in whether they are allowed to drive unassisted. The median duration of visual decline was 22 to 33 days. Next slide. So I want to make sure that we talk about some of the uncertainty and controversies. There is an ongoing concern about treatment-related deaths, especially with cilta-cel. Again, for cilta-cel, the data is immature and preliminary. And so as I think you may recall, when we looked at, for example, progression-free survival or overall survival, we were only able to produce greater than a certain number because the data is premature and preliminary. And so there's additional uncertainty about cilta-cel compared to the other treatments. And the third point that we wanted to raise was that for many of the other, previously for myeloma, the progression-free survival and to OS ratio for all the other treatments for myeloma has been remarkably consistent at around 2.5 to 3.0. The PFS to OS ratio for belantamab is an outlier compared to all other myeloma treatments. And I think additional studies are needed to see if this is actually a the outlier status is a reflection of the truth or is actually a spurious finding, because ultimately all of these studies are relatively small. And so I think the chances that any of these numbers may change with additional experience is significant. Next slide. So I also want to talk about some potential other benefits and contextual considerations. So I think the primary benefit that we have to consider is that anti-BCMA therapies work through a novel mechanism of action. And so making it more likely that these treatments are going to benefit patients who are really refractory to all other current treatments. The harm, the contextual harm that I would like to highlight is the fact that these treatments may worsen disparities, and specifically previously medications and treatments with high cost and high side effects. And I think these treatments would certainly qualify under both of those descriptions as being costly with high side effects and requiring treatment at specialized centers have often worsened disparities. And so I think this is something that we need to consider. Next slide. So I want to just briefly review some of the comments that have been received. So for belantamab, the ocular toxicities are temporary and reversible. And so I think it seems like in the overwhelming majority of patients, and it's pretty close to nearly all, the visual impairments that we've seen get better once the drug is stopped. And so I think that is definitely something to keep in mind. The published data, as I briefly mentioned, for ide-cel and cilta-cel rely on an as-treated analysis. The intention to treat analyses are more likely to lead to unbiased comparisons to usual care, and that's why we highlighted the intention to treat numbers. And finally, I think it's important to stress that right now there is insufficient data to compare anti-BCMA treatments to each other. Next slide. So I wanted to kind of go and summarize the evidence ratings for these treatments. So first, for triple or quad refractory myeloma for patients who have received 3 or more prior lines of therapy, the comparison of ide-cel versus usual care, we settled on a rating of B+, which means incremental or better with a high certainty of at least small net benefit. And so we're saying that ide-cel compared to usual care is better, maybe a little bit better or maybe a lot better, but we have high confidence that it is better. We ended up with the same evidence rating for cilta-cel versus usual care of B+. So again, incremental or better with high certainty of at least small net benefit. For a comparison of ide-cel versus cilta-cel, as we mentioned, we felt like we settled on insufficient as the evidence rating, and so we don't feel like there is enough data to compare those two. And for the population of triple and quad or pentarefractory myeloma with 4 prior lines of therapy, belantamab versus usual care, we settled on a PI rating, which is promising but inconclusive, with a small likelihood of potential negative net health benefit or harm. So our best estimate is that the belantamab versus usual care is equivalent to potentially slightly better, but we can't rule out a small net harm also. Next slide. So again, summarizing the results for the CAR T therapies for myeloma, ide-cel and cilta-cel, the current evidence suggests a high likelihood of at least small benefit compared to usual care, with a possibility of substantial benefit. For belantamab, current evidence suggests equivalence or small benefit compared to usual care, but cannot exclude the possibility of small net harm. So I managed to, next slide. Okay, so I managed to end 3 minutes early, so I'm happy to take questions at this point.
Thank you very much, Dr. Lee. I'm going to keep an eye on if anyone has their hands up. And we have about 5 to maybe 6 minutes time for a question. Let's start with Dr. Bora.
Yeah, hi, thanks for the presentation. So my question is with regard to the usual care basket. So it seems the usual care could be combination of any lines of therapy, and it could be given what I have read, it could be really kind of heterogeneous. So when we say when you put usual care in one basket, I mean, have you taken into consideration the various say, I don't know, combination A, B, C might have very different effect versus combination A, C, D. You see what I'm saying? So how was that heterogeneous nature of the usual care was taken into consideration when you compared each of these 3 drugs with usual care?
So that's a great question, and it quickly gets into the weeds. So I would say that we were relying primarily on the Mammoth Study, and this was a study, a retrospective study of similar type of patients, and they had a long list of how frequently patients were getting which type of medications. And so we did a literature review and relied on the Mammoth study to kind of get proportions of, these are the proportion of patients who would be getting this sort of regimen versus this other sort of regimen. And for the modeling component that Dr. McQueen will talk about shortly, I think we use those weights to characterize the usual care group. I don't know, did that answer your question?
Sure, okay, all right, thank you.
Thank you for that answer. Greg?
Thank you very much, Say. That was really an outstanding presentation, excellent summary. My question relates to belantamab, and my understanding in reading the documents is that many patients who initiate therapy drop out, they discontinue the medication, and that really is an important consideration, I think, in coming down to a bottom line evaluation. And I wondered about this PI assessment, if it's truly promising, if so many patients are discontinuing therapy. And I wonder if you could comment about that.
Yeah, I think the hard part is how different some of the data, some of the outcomes are. So I think the most favorable perspective for belantamab is to look at the overall survival. And so if you look at the overall survival, it does seem to be several months, quite a few months greater than in usual care. If you look at some other outcomes like overall response rate, I believe it was 32% versus 28% for usual care. And so and certainly if you look at health-related quality of life, doesn't really feels like there's much of an improvement there. So I think there is, the other thing to, I guess, if I was thinking about what are the most positive things for this medication, is that this is a heavily pretreated group that has very few other options. And so I think suggesting that as a single agent to be able to have this much of an effect, for me, much of the promising is going to be actually is a little bit outside of the scope of our review, which is I think the interest is going to be in how belantamab can be combined with other treatments. And I think that those studies are currently being done, and outside of the scope of this review, specifically with the promising part, I do think there is definitely some data elements suggest that it is a little bit better than usual care, and I would point to the overall survival numbers as the primary ones there. Does that answer your question? It's a long-winded answer.
Yes, thank you very much, Say.
Thank you. I see Stacy.
Thanks so much for that presentation. I also have a question about belantamab and the ocular toxicity. So you mentioned that it's reversible and temporary, and I just wondered, do people have to stop taking the drug in order for the toxicity to reverse itself? So do they have the impairment the whole time they're on the drug?
So yes, and I think this is something that our clinical experts may be able to talk in greater detail, but it is actually, with the approval, there was a very kind of clear guidance on exactly what needs to be done. You need a baseline ophthalmology evaluation, and then regular ophthalmology follow-up. And then when they start seeing things, depending on the degree of ocular toxicity, they're going to hold the medication or decrease the dosage. And so this is something that is usually, so this is something that there is a protocol to deal with. In terms of, I think the last thing you mentioned was that this is something that happens for the duration. The median duration, I think I listed, was around 22 to 33 days. And so it is reversible, but it is not something that goes away immediately. And I think it is something that I think for me, one of the things that I was thinking about was that these are patients with a fairly limited life expectancy, and to have visual impairment, significant visual impairment for a substantial proportion of that limited life expectancy, I think is something that played into my concerns about that this could actually lead to net harm.
Thank you.
We're gonna have the last question from David, and I'll save my question for later. David?
Thank you. My question has to do with stem cell harvesting. Autologous stem cell transplant, I believe, is still standard of care early or late for multiple myeloma patients. If a patient harvests stem cells and gets enough, can those same cells be used for these drugs we're looking at, or is it a different cell that you isolate? Do you know what I'm asking?
So I have, the first thing is I just got a text saying that this actually, that this part of the meeting should be focused on answering questions from the panel. So can I get guidance from somebody from Icer on whether whether we should try to, whether David will have a later time to ask the question?
No, it's fine. It's absolutely fine. Comments or questions are fine. We have to move on pretty soon, and this might be something easier for one of the clinical experts to answer anyway, but why don't we go ahead and address it since it's on the table?
Yes, I would love help from the clinical experts on this one.
Sure, I'm happy to answer that. So currently, we cannot use stem cells that have been harvested for an autologous stem cell transplant. In the leukapheresis, the cells of interest are lymphocytes, whereas for autologous stem cell transplant, we just collect all these CD34 positive cells. I think there is work being done on how to utilize harvested stem cells. So in the future, this might be something that can happen, but unfortunately, currently it means that even if patients have had harvested stem cells, they still need to undergo yet another leukapheresis.
Thank you. My question is answered.
Excellent. Thank you all. And thanks, C, for this excellent presentation and really great discussion. And with that, we're going to move to the presentation of evidence and economic modeling with Brett McQueen.
Thank you, Reem. Can you hear me okay?
Yes, we can.
Great, thank you. I'm Brett McQueen. I'm an assistant professor at the University of Colorado. Next slide, please. Just want to acknowledge our team from the university as well as the team from Icer. It's always great collaborating with Icer, and we really appreciate it. This was a challenging but very rewarding project. Nothing to disclose in terms of financial support other than from Icer. Next, please. So our objective for this was to assess the lifetime cost effectiveness of ide-cel, cilta-cel, and belantamab separately compared to relevant comparator treatments. Next slide. Next one. Yeah. So this is over a lifetime as typical modeling in oncology from a healthcare sector perspective, and we do provide a modified societal perspective in the supplement. The results are fairly similar, and I'll get into that in a little bit. The cycle length reflects typical treatments of usual care in this space, where they range from about 3 weeks to a month of administration. And our outcomes are total costs, life years, quality adjusted life years gained, and equal value of life years gained. And then we have incremental cost effectiveness ratios with those outcomes in the denominator. We also offer an alternative cost effectiveness metric, cost per month of progression-free survival gained. Next slide. So this is a model schematic of the structure. On the right-hand side, you're going to see patients starting in alive and responding to treatment. In other words, progression-free survival. They can pass to alive and not responding to treatment or progressed disease, and then death is all cause or disease specific. You'll see on the left-hand side, and I'll make this point numerous times throughout the presentation, we aren't comparing across interventions. We're comparing to each therapy or intervention to the respective usual care market basket. I know there was a question earlier about the market basket. If you're interested in looking at the exact or the specific therapies, you can find that in supplement tables E.2.3. and E.2.2. I think I said that the wrong way. In the case of the CAR T therapies, were compared to triple or quad refractory regimens. And in the case of the CAR Ts, we include this upfront decision tree. So Dr. Lee talked a little bit about the on-treatment analysis, and the model we're actually accounting for intention to treat by following patients from leukapheresis all the way through CAR T infusion. And in that way, we're able to assign the comparator risks, benefits, and costs for those that weren't infused, so that in the end, it's sort of an average. In the case of belantamab, as Dr. Lee discussed, it's more of a weighted average of triple, quad, or penta refractory regimens. Next slide, please. So you'll see two tables here. The top one will start with that triple or quad refractory multiple myeloma population. And again, I want you to focus in on each individual comparison to the triple or quad refractory subset from Mammoth. And we were able to, Mammoth was a great study for this. It's real world, and it was able to stratify overall survival curves by these different subgroups of refractory status. And you'll see in the top table, generally across categories such as age, refractory status, median prior lines of treatment, they're generally comparable to Mammoth. On the bottom table, you'll see the triple, quad, or penta-refractory multiple myeloma population. Again, across age, refractory status, median prior lines of treatment, they're generally comparable to what we're assuming is a weighted average of the triple, quad, and penta-refractory population. Next slide. So there are some key model assumptions. Anytime we're in a space which is driven largely by survival, we're going to be making extrapolation assumptions beyond the observed data. And so it's important to understand that some of the extrapolations may differ depending on the distributional assumptions. You can find some of these in the supplement for figures and different assumptions around the Icers. I talked a little bit already about Mammoth, but it's an observational study from academic centers around the country. And we paired that up with a conference proceeding that really identified therapies used in this population. And again, you'll see those in the supplement where we list all the therapies and then the prices and the dosing, et cetera. For CAR T therapies, all patients, or excuse me, a proportion of patients received at least one full single course of therapy. This is an update from the draft report where we include a proportion or a smaller proportion of that originally treated population who were assigned retreatment. And in the base case, this is very important that they were assigned the full cost of CAR T, as well as all the other resources used. So managing cytokine release syndrome, other adverse events, inpatient settings, et cetera. And I kind of already spoke to the intention to treat, where for those that were not infused, they received the comparator treatment benefits, risks, and costs. Next slide, please. So we'll start with a few different slides on key model inputs. Obviously, survival and mortality are the most important. And you'll see on the far left for ide-cel, the gain in progression-free survival of about 9 months and overall survival about 19 months is higher than what we observed in the triple or quad refractory population, that sort of middle column there of about 3 and 9 months. In the case of cilta-cel, we're going to stress that the evidence is quite preliminary. We were left with one progression-free survival curve where the median had not been reached, and obviously thus the median for overall survival was not reached either. That is also compared to the triple or quad refractory population. On the far right, 2 columns, you'll see belantamab. This is a very interesting part that Dr. Lee stressed, and it really comes down to some of the modeling assumptions that we're using. And we provide some scenarios around this that we hope will kind of shape the discussion a little bit more. But you see that belantamab has this almost 3-month median progression-free survival gain. And then in the weighted average of triple, quad, and penta, and if you go to the actual paper itself, you're not going to see these numbers necessarily. It's because we are weighting the outcomes from these 2 populations, triple and quad, and then penta refractory. So Dr. Lee went into this a little bit, but the typical evidence in this space, at least based on meta-analyses, have suggested for every 1-month gain in progression-free survival, there's about 2.5 to 3 months gain in overall survival. So right away you see, even though the median PFS is quite similar, overall survival is quite a bit dramatic and almost I think Dr. Lee used the term outlier for belantamab. Next slide, please. These are key model inputs for intervention costs. This is another update from the draft report. So in the draft report, we assumed prices that would meet threshold such as 100,000 per quality adjusted life year. With ide-cel recently approved with a price of about 420,000 per infusion, we then used that and updated the model results. So that's part of the reason why the draft report and the final report are different in that sense. In the case of cilta-cel, it's not approved yet. We did assume the same price as ide-cel, and that's why it's listed as an assumption for now. An interesting part about belantamab, the list price per 100 milligram package or vial is about 8,200, which comes out to be per month anywhere from about 20 to 30,000 per month, which is quite similar to comparator or usual care therapies. You see that range is between 20 and 25. That's the range of more of the mean. We actually, around the one-way sensitivity analyses, have a broader range that moves from about 17 to 35,000. Next slide, please. So I want you to focus on the top part of this slide because that's really where there's differentiation between the different treatments. Prior to and during therapy administration, ide-cel had about 92% infused from the trial, 22% who were retreated, and then intensive adverse event treatment in inpatient settings. That ranged from about 6 days in an inpatient setting up to 24, which is consistent with previous CAR T evidence that the median time is about 15 days spent in inpatient settings. In the case of cilta-cel, 86% were infused. We didn't have a proportion that were retreated, so that's an assumption that we used from ide-cel. And you'll see in the tables and the evidence summary, as well as my presentation here today, that the cost difference will sort of play out between the two. It's largely a function of who was infused, who was not infused, and then who was retreated. So again, another reason why we don't want to compare across CAR T products. In the case of belantamab, there were examinations. I think Dr. Lee talked a little bit about this at baseline and prior to each dose. We include those resources in the model. Stage 2 and stage 3 are consistent across all comparators and treatments. So we have typical monitoring done in multiple myeloma. And then in terms of progressed disease, we apply the same number of subsequent cycles. Next slide, please. So in order to calculate quality adjusted life years, we need utility scores. And the recent KarMMa study actually did provide us with differentiation across progression-free on and progression-free off, as well as progressed disease. And this is something we learned during engagement efforts with patients, where there is a benefit from not being on dexamethasone, from not being on some of these triple or doublet therapy regimens. And so we tried to express that in the model and actually quantify it in terms of differences in utility. And so that's why you see there's a difference between on and off therapy. And then in terms of progressed disease, there's obviously a difference as you move to that health state. We also included disutilities for other adverse events that varied. And then for grade 3 or 4 CRS, we assume a utility of 0 for a duration of 15 days. That's consistent with the prior CAR T report from Icer. Next slide. Thanks. All right, so how does this all shake out in terms of results? Well, on the far left column, we have intervention cost. And you might notice, why is the intervention cost greater than the list price? And that's because we're including things like retreatment. Not everyone was infused, so you can't just simply multiply it by that 22 or 1.22. And so that's all adjusted up front in that decision tree. Other non-intervention costs include progressed treatment costs, costs for monitoring, physician visits, adverse event management, et cetera, for a total cost of about $650,000, as compared to usual care of about $275,000. Now that extra cost comes at an extra benefit. And you can see we've provided about 4 different outcomes: time spent in PFS state in terms of months, quality adjusted life years, equal value of life years gained, and life years. For those of you not familiar with equal value of life years, we're still incorporating baseline health-related quality of life. We're still incorporating those quality of life measures. However, when patients say ide-cel has an extension of life, as you can see here, during that extension of life, if patients happen to pass to the progressed disease state, we're not decrementing their health utility for passing into that progressed disease state. We're assuming a constant or average estimate from the US population. That's why it sits right in the middle of the quality adjusted life year and the life year gained. Next slide, please. So for cilta-cel, I'll stress this is preliminary evidence. Again, we see that the intervention cost isn't exactly that list price. And the total cost, as I mentioned on the previous slide, the total cost is a function of both those infused, those not infused, and any of the additional sort of other non-intervention costs like I just talked about. So that's why the cost is different. This also comes at an extra benefit, time spent in progression-free, additional quality adjusted life years, equal value of life years gained, and total life years. Next slide, please. Now, in the case of belantamab, I tried to stress early that progression-free is very close to the usual care basket. And it's also very close in terms of price. So you're kind of seeing this play out in terms of the results, where the total cost is about $250,000 versus $218,000. We can't share the time spent in progression-free state given submission of academic and confidence data from GSK. The additional quality adjusted life years, equal value of life years, and life years gained are displayed on the right three columns. Next slide. Okay, so these are the base case incremental results. So we'll start with the first row. Remember, we're not comparing across these. These are all compared versus usual care. ide-cel comes in with cost per quality gained of about $320,000, cost per equal value of life year gained of about $280,000, and so on for cost per life year gained, and then cost per additional PFS month gained. In the case of cilta-cel, again, we'll stress this is preliminary. A cost per quality gained of about $150,000 and a cost per equal value of life year gained about $130,000. Case of belantamab, this will be very interesting. Dr. Lee talked a little bit about some of the uncertainties that will be reflected in the sensitivity analyses. And you can see it sits right at that 100,000 per quality adjusted life year mark, and then just slightly lower for the cost per equal value of life year gained. Next slide, please. So this is one of 3 different one-way sensitivity analyses we did. So obviously progression-free survival is a big driver, and as expected. So at higher levels of benefit, we would expect the Icer to minimize slightly, but still higher than commonly cited cost effectiveness thresholds. And then at lower progression-free and overall survival, we see the Icer increases. Now, just to be clear, when we change progression-free survival in the model, we're assuming the same relationship as we've observed in the trial, so that if, say, patients have more of a 12-month progression-free survival, that would be multiplied by 2.5 or 3. In the term, the other key driver was unit price of the comparator market basket of therapies. And at lower prices, closer to 17,000, you see the Icer increase, still above commonly cited cost effectiveness thresholds. And then at higher, that higher range, it reduces slightly, again, still above commonly cited thresholds. The other estimate, which might be very informative for future research, is utility of progression-free survival, both on or off treatment, was another key driver. Next slide, please. So in the probabilistic sensitivity analysis, we're repeating the model calculation while varying multiple inputs instead of doing a one-way sensitivity analysis where we only fix one input and then see the resulting impact on the Icer. So in this case, you'll see both ide-cel and separately cilta-cel start becoming greater than 0 or 1% at higher thresholds at 150,000 per quality. For belantamab, this might be the most interesting finding on the table. You saw that the Icer was 98,000 per quality, yet in multiple and thousands of iterations, only about 50% of those calculations met the 100,000 per quality threshold. So remember, we're right on the edge of the price. We're right on the edge of that survival. It's a very interesting finding from a modeling perspective. Next slide, please. Okay, so we have a couple of scenario analyses. I did mention that there was a third on the modified societal perspective. They're very similar to the base case. And so we're highlighting the two that we feel like are really informative for the evidence summary and the voting. And the first one is assuming no additional charge for CAR-T retreatment. That said, we're still including the cost for adverse events, for cytokine release syndrome. We're not minimizing that cost from a hospital perspective. We're just assigning, removing the CAR-T infusion cost. And you see for ide-cel, the cost per quality gained is about 250,000, cost per equal value of life year gained at 217,000, still above commonly cited cost effectiveness thresholds. Again, in the case of cilta-cel, this is preliminary given the limited evidence. Cost per quality gained of about 110,000, and then cost per equal value of life year gain just breaks 100,000 per quality. Next slide, please. So this is the interesting belantamab scenario analysis, where if you recall, for every 1 month gained, we're in between 4 and 5 months of overall survival gained. And so in this scenario, we actually adjust the relationship to be consistent with prior meta-analyses. And we see that in that case, belantamab is more costly and less effective. And so we've stressed that that base case estimate is fairly unstable to alternative assumptions or sensitivity analyses. Next slide, please. So there are a number of limitations to note. First, given a lack of quantitative indirect treatment comparison evidence, we should use some caution when interpreting these. You can think about the cost effectiveness estimates actually changing both ways, depending on if the population that we're comparing to would be more severe or less severe. Specific to cilta-cel, I've said this numerous times, interpretation should be noted as very preliminary and further evidence is needed. I already mentioned a little bit about survival extrapolations. You can see how those change in the supplement. Next slide, please. So first up, on some of the comments that we received, we did underestimate the observed survival for ide-cel and the comparators. We acknowledge that, and we did fix it in the report, and now we're slightly overestimating the observed tail end of the KarMMa trial. In terms of the update, I already mentioned the draft did not include the retreatment of CAR-T therapies, and that's new evidence that came along mid-review. And then we made some slight adjustments to utilization and monitoring around belantamab's vision complications. Next slide, please. So to conclude, findings suggest the CAR-T therapies provide clinical benefit in terms of gains in quality adjusted life years and life years gained. Ide-cel meets commonly cited cost effectiveness thresholds with discounts greater than 37%. You can see that's really based on that threshold of the equal value of life year gained. The preliminary evidence on cilta-cel, which needs further evidence, meets commonly cited thresholds given this limited evidence. Then findings for belantamab suggest the current list pricing is within commonly cited thresholds. However, I talked about how unstable that was. And so it's important that we're looking at the inputs and the assumptions that we've used and interpreting them in context to those, which includes baseline survival, price of other therapies, et cetera. I think we have questions now. Thank you.
Thank you very much, Britt, for simplifying a very complex body of work and presenting very clearly. We appreciate it. Again, I'm going to keep my eye on the chat or the raise hand from anyone from the Midwest CEPAC. Excellent. So, Brad?
Hey, Brett. A nice presentation, a lot of work. I guess I just had a question that's maybe for the clinicians and the modeling. So this is kind of a thought exercise. So in the clinical evidence table, we say that, Icer came out with the recommendation that's inconclusive or insufficient to compare ide-cel and cilta-cel. In the modeling, like in the sensitivity analyses, progression-free survival clearly is the one that drives, the value here. But when you look at the ranges used, the most optimistic one used for ide-cel was 12 months PFS. The worst case estimate that was used in cilta-cel was 13 months. So there's not even coverage, it's like the modeling, there's no overlap in that progression-free survival. And I'm just trying to, maybe this is a thought, kind of how to wrap our heads around where we're kind of in the clinical evidence, we're saying it, we can't tell a difference, but in the modeling, there's a difference. And even in the modeling, even in the most extreme sensitivity analyses, we're still saying that there's no overlap between the two products. And also be interested to hear from the clinicians, kind of what is, and I don't work in oncology, what might be driving, differences between these two regimens that are largely the same, but, and I know the cilta-cel evidence is, we don't have a whole lot of data on that, but, is this a real finding or is this an artifact of kind of, we're only running about 150 subjects to these trials and we're seeing that. So lots of questions. I don't know if we have time to answer them all, but I'm just kind of laying out what's going through my head as I think about this evidence.
Yeah, thank you, Brad. And I'm going to let the clinical experts speak up about mechanism and differences and why we might see that in the trial. From a modeling perspective, and a lot of what we saw early on with ide-cel was a little bit more optimistic, and we're seeing the same with cilta-cel. And it's tough to use the word optimistic. I mean, and I think Dr. Rajkumar said it best last week when he mentioned that the truth is probably somewhere in the middle. And even though there's no overlap, it was very challenging from a modeling perspective. And that's why we stuck with this is preliminary evidence, and we really need, further beyond phase 1 trial evidence to make more conclusive determinations, at least around modeling. So I'll stop there because I'm going to soon get out of my comfort zone and say something wrong.
Any of the clinical experts would like to comment on that? I think it's a key question that's in my mind too.
So, there are, unlike standard myeloma treatments where you just pick one off the shelf, there's a lot of factors that go into who was able to get ide-cel or who was able to get cilta-cel. And, ide-cel started early, and so there's everything from which centers had the trial open to which company was more strict about patient eligibility criteria and any leniency with regards to how much bridging therapy or what we can use. There are many factors that go to determine whether even a triple refractory patient could be quite different going from one trial to the other. So I think in this particular case, I wouldn't make cross-trial comparisons. I totally hear you that says like even the worst case scenario and the best case scenario don't overlap. But I truly think that I was, as you were presenting, Brett, I was thinking like one analysis would be to merge the two and give us what the data would be, because that's probably what will happen in real life if we had the same patients again, even if the trials were repeated.
Yeah, and the only thing I'd add to, Brad, is that remember, we're using placeholder price. We're using placeholder retreatment. The upfront number of who was infused, the number that's different between the trials. And I tried to explain that total cost will be different because of that. So there are just so many differences that it's hard to compare. And I think, Dr. Rajkumar, to address your point, we can sort of see the overlap between the two at that upper progression-free survival estimate and then at the lower for cilta-cel. And so we didn't want to necessarily present that because we don't want to compare across products, but hopefully you can visualize what that would look like just simply from the results. But again, we don't want to compare across CAR Ts. I think that's the main point.
It's easier said than done.
Yeah, so I'm going to ask a version of the same question, which is, if we have decent results in the ide-cel and provisional results on the cilta-cel, our best guess is maybe the results are somewhere in between. But should I toss out the cilta-cel results because, or not toss out, but give them less weight because they are so provisional at this point? Like, how am I supposed to think about this? Because it looks to me like we've got two very different pictures of these drugs. On the one hand, they're either highly cost effective or they're not cost effective, depending on which data set you pick. And I guess I'm struggling to figure out how to merge these two very different sets of results. And is the quality of evidence sufficiently greater with ide-cel that I should weight that much more?
I'm going to stay away from saying the quality of evidence and then your interpretations, and I'll stick with the evidence for cilta-cel is very preliminary. We relied on one progression-free survival curve. And then we, in a sense, extrapolated that the same observed progression-free to overall survival assumptions would hold for cilta-cel. So you can think of that as preliminary. You can downweight that if you want, but that's not my call. That's your call. It's more about the number of assumptions that we actually had to make to get a cost effectiveness estimate alone. We're pretty great in that. And ide-cel is just further along in terms of the trial development, in terms of the approval. I'm not going to say there's a difference in necessarily in quality if they were compared at the same stage, for example. They're just at different time periods. And so I think it's up to you to decide how you would weight that. But hopefully I didn't punt too much on your question.
This is Say. If I'll jump in there and I guess not say anything qualitatively different than what Brett mentioned. I just think that the data for cilta-cel is earlier. And therefore, I think, when you first said, so do we just toss out the cilta-cel data? That feels, that makes me uncomfortable. I think the data is preliminary. And so I think when we talk about, do we weight the ide-cel study data more, that seems much more reasonable. But ultimately, it is, I think this is what, as for the clinicians in the room, I think this is what we are often trying to do as clinicians, is like we have imperfect information, but we still, with the choice of not making a decision is a decision in itself. And so we are trying to use the best imperfect information that we have, even if some of it is preliminary. And it's a very complex algorithm to try to figure out how much you weight each one. But I don't think you throw out the data. It's just that it's so early that it just doesn't count as much. And what that means is the final result, the reason we don't count it as much is that we've, in the past, this preliminary data, when the data matures, there's actually a fairly wide range of what we may actually end up thinking, the conclusions that we end up taking from this study once the data actually becomes mature, but we don't have that right now.
Well, thank you. Let's see, Albert?
Yeah, Say and Brent, great talks. I just want to make sure I understand why we have the Icers that we have with ide-cel and cilta-cel. And this is just my attempt to simplify what, combining the clinical evidence with what happened probably in simulation. My understanding is that ide-cel actually has pretty profound quality of life improvement during the course of the trials. Less fatigue, and all that translates into probably a change in utility. And it has some benefit in progression-free survival. Cilta-cel, I remember from the evidence, it's actually minimal. It doesn't have the same quality of life benefit, but does it have more of an effect on PFS? I'm just trying to understand how cilta-cel ends up with the lower Icer compared to ide-cel, even though ide-cel has a larger quality of life benefit.
Yeah, thanks for the question. So I think I had mentioned before that, if you actually assumed the same proportion that were infused versus not infused, you get the same essentially total costs, or very, very similar. So in terms of the intervention cost itself. So that's already one that's a differentiating factor, just simply the proportion that were infused. Because we're not only accounting for the proportion infused, we're also assigning the benefits, the risks, the costs of the proportion that wasn't infused. They pass to the comparator therapies, right? But both use assumptions around health utility that if you're off treatment, there's more of a benefit, and that's in the progression-free state. So that's the utilities are very consistent across, and they are derived from the KarMMa study, rather than evidence from cilta-cel. So again, the number of inputs that we're using in terms of the assumptions for ide-cel to cilta-cel are pretty great in this exercise. All of that said, the driver for the difference in the Icers is largely the progression-free and overall survival estimates. And remember, I'm going to remind you one more time that the overall survival was an assumption that we extrapolated based on the available progression-free survival curve. So we don't have a overall survival curve yet. We're still waiting for that evidence to come out. So a lot of assumptions made, a lot of overlap between ide-cel and cilta-cel because we were essentially forced to get there with those assumptions. So I don't know if that at least helps frame it a little bit more for you, Albert, or?
What happens if you do the, have you done also an analysis where it's received treatment, not intention to treat?
In the model, we certainly could do that as a possibility. But in terms of intention to treat, I mean, we'd rather, because those patients in reality will pass to a new market basket of therapies, we really wanted to stick with ITT. So
just to make sure we stay on time, we're gonna have one more question, hopefully a short one with a short answer, and then with Timothy, and Ellen will try to get your question later during the day. Timothy, go ahead.
Yeah, thanks. I think my question will be short. You mentioned in the limitations, and it's a great report, Brett, thanks, that the survival extrapolations are sensitive to the parametric models. And looking at the curves, it did look like that was the case. And I think you made smart decisions about the parametric models, but could you speak to that as well, about the sensitivity to that?
Yeah, and I think, so early on, I'm sorry, Reem, I'm going to try to keep this short, but early on in the draft report, we did acknowledge that we underestimated survival for ide-cel and the comparator therapies, and largely because, you know, for all of the talk of between PFS, between the two CAR T therapies, the benefit is pretty substantial over usual care. And it's hard to figure out in the last CAR T report, you know, we had this actual plateau happen. We're not seeing that yet. So a lot of these assumptions at the tail are really driving some of the results. And if you look at, for example, table E.4.3, you'll see that if we use different assumptions on the survival extrapolation, we get remarkably different results. And so what we tried to do is be completely transparent as we possibly could and say, yes, these will differ, but also get the best fit possible to the observed data. And that would be our base case. So that was kind of our reasoning behind that.
Well, thank you very much. With this, I think it is our time to go to the public comment and discussion. Again, I'm going to ask everyone who will present. We have 2 manufacturers and 1 public comment.